Okay, this is the last day, and this morning we're starting with Sharon Thompson-Shiel, one of the organizers. She kindly agreed to speak at the last minute after last-minute cancellation. So we're very happy to have her speak here. She said she had this talk already ready, prepared for this occasion. So thank you very much.
Thank you. Yeah, so I am not Todd Griffiths. Sorry to disappoint people who wanted to hear about deep learning, just shallow learning this morning. Let's see. Does this work? No. Yeah, now it's working. All right. Okay, so this is the 11th CAOS. I first came to CAOS in April 2008 at the second CAOS. So when I was asked many hours ago to give this talk, I reflected back on the past 10 years. Here is what was going on in the world back in 2008. So there was bad things going on. There was some good things going on, and this different picture in both of these today. And speaking of pictures, I found a picture of me, much younger, 10 years ago. But I chose this particular picture because you can see this very high-tech piece of technology. I am holding here my phone with its actual keyboard on it. And I chose this picture because at the time that picture was taken, I had just published, along with two of my students, a book chapter that had this figure in it with reference to a telephone. And so the way I want to start today is by taking you down a little stroll through memory lane with me about what was going on in the field at the time that this figure was produced. This figure describes a theory that was first introduced by Allport many years before, that our knowledge about concepts is distributed across the sensory and the motor systems through which we acquired the knowledge of those concepts. And so that's what's being illustrated here, where what we know about the action of a telephone and what it looks like, how it sounds, nothing about how it tastes, and so on. So this idea predates the advent of functional neuroimaging, but when functional neuroimaging came on the scene and people started thinking about what they could do with this new technology and with the analyses that were present at the time, this became a hot area for applying neuroimaging techniques to test, or so we claim, to test some of these ideas. So let me give you an example of how these sorts of experiments worked. You would have subjects do things like look at colors versus achromatic stimuli, or make judgments about chromatic versus achromatic stimuli, and you would subtract the neural patterns that were evoked when subjects were looking at the stimulus on the left. You would subtract from that what was evoked when subjects were looking at the stimulus on the right, and you would see what areas responded differentially. And then you might, in a different group of subjects, or when you got really fancy, even in the same subjects, you might have them do things like look at a picture of an object that has an associated color and report what color that is. And then you might subtract from that neural pattern the activity when subjects were naming the concepts, so saying that this is a bulldozer. Or maybe you would compare saying whether an eggplant is purple to saying whether or not you throw a football. Okay, and again, you would subtract the images that were evoked in those conditions, and you would see things like this. So you would find localized places in the brain. What you're looking at here is the ventral surface of the brain, so turned over, and this is the left hemisphere, the mid portion of the occipital temporal lobe here. So this is an area that responds more when people look at color versus achromatic stimuli. And then here, this is an image comparing when people are saying that a bulldozer is yellow versus saying that the picture is of a bulldozer. And then you would observe that these two places were kind of near each other. And then you would conclude that our knowledge of the color of objects depends on the same areas that support color perception. Okay, so that's a sample result, a sample inference that was drawn. You could have a whole talk on, what it means to say this is close to this, and what it would even mean to overlap. And it wasn't just color. There was work looking at, for example, tools and premotor cortex. Here's the color paper I just had. Manipulation. We heard a little bit about manipulation yesterday. You have size, color, sounds. Color, form, function, so on and on. Taste, this had lots of different sensory areas. Cinnamon activates olfactory brain regions, and so on. And so all of these kinds of studies, which many papers from my group are part of the collection as well, were taken as evidence that our concepts were distributed through these sensory motor systems, and therefore that our concepts are grounded in sensory experiences. All right, so that was CHAOS 2. I think many of those results, stand today, and I'm not saying they're not important and they haven't contributed to the field, but I have been wondering recently whether or not they tell us anything interesting about concepts. And when I was in graduate school, one of the first things I learned is that concepts aren't just lists of features. They're not just lists of properties or even probabilistic properties. And so this whole notion of breaking the concepts apart into these features, I think, was really driven by the tool that we had. Fmri was really good at breaking things apart. That was the whole thing. I mean, how many times have you heard the phrase, we're going to carve nature at its joints? But carving concepts at their joints might be doing them a disservice. And so lately what I've been trying to do with my group is think about ways, driven by new tools perhaps, and old ideas, to think about putting concepts back together. And that was the Humpty Dumpty reference. So what I'm going to present today are some baby steps in this direction. Hopefully, if I'm invited to speak, or if there's a cancellation 10 years from now, I will have much more progress, much more to say. And so this is going to be some baby steps to give you a sense of the directions in which we're moving, thinking about integrating concepts. The first part of the talk, I'm going to discuss some work done by Marc Coutanche when he was a student in my lab. I don't know if he's still here. Yeah, he's here. So you can direct all of your questions about this to him. And Marc's now a professor at Pitt. And so I'm going to describe some work we did looking at parts, looking at two particular parts of the visual system, but trying to think about how information is transformed when you actually form a concept with those parts. So that's the first bit. Then I'm going to talk about a recently published paper where I still stay grounded in the visual system with a visual part, the shape of an object, but I start to look at how that can become integrated with abstract information. And I'm going to present here what I think you'll find to be a surprising result. I found it to be a surprising result that, again, looks at how information is transformed and how information is changed as we learn about concepts. And then at the very end, I'm going to describe some very new directions that we're moving in the lab, looking at not how we combine a visual feature with another visual feature or a visual feature with an abstract feature, but how we take one concept and combine it with another concept to create new concepts. Because that's what's interesting about our concepts, is that we can work with them, right? And so I think this focus on decomposing them into their parts, we've lost how powerful they are and what they do for us. And so that's the direction that we're going. Alrighty. So, from visual features to concepts, and as I mentioned, this is the work of Marc Coutanche when he was in my group. So what Marc did is he had subjects imagine, think about, and the task was to search for one of these four concepts. They didn't know exactly what the picture would look like. There were many different pictures of carrots that they might see. But it was always an exemplar from one of these four different fruit and vegetable categories. The subjects were cued which one they should be looking for. And then they would see these blinking screens of just a Gaussian noise patch. And then eventually there would be an object would appear somewhere in the display. and they would have to say whether it was what they were searching for. And what we did is we analyzed the data from the period of time when there was nothing on the screen. So really what we're after here is the thought about a tangerine and not the patterns that are evoked when looking at a tangerine. The way Mark analyzed these data, he did a number of different things. But to look for the parts, what he did is he asked whether or not he could distinguish the neural patterns that were evoked when people were thinking about the orange stimuli from when they were thinking about the green stimuli. Now, if you just trained a multi-voxel pattern classifier to make this distinction versus this distinction, there's lots of ways in which those stimuli differ from one another. So instead what he did is he trained it to make the tangerine-lime distinction, and then he took that model, that classifier, and he said, okay, now when I look at the neural patterns that are evoked by these stimuli, can I predict what people are thinking of? And the idea is that if you can distinguish the tangerine from the lime, but now generalize over this big transformation in shape, you know that you have a pattern that's coding color. Okay? And then vice versa for making the distinction based on shape, and can you now generalize to the other colored stimuli. And in doing so, he found areas that were coding color in a way that generalized across shape, and coding shape in a way that generalized across color. And they were the likely suspects. So he found regions of lateral occipital cortex where he could classify the other colored items based on their shape. And then he found regions in what corresponds to V4 where he could discriminate color generalizing over shape. Interestingly, there was also a marginal response to discriminate on shape generalizing over color. And what I find very interesting about this is that it can't be an integrated source of information, because if it was an integrated code, you wouldn't be able to do the generalization over the other stimuli. So I think that's what's so powerful about that generalization test. So you might have two commingled population codes here. Okay, so now we have some parts. We have some object features. Then Mark asked, are there regions of the brain where you can train a classifier to distinguish amongst the 4 different concepts? So again, this is all when they're thinking about them. And so they're not experiencing color, they're not experiencing shape directly, and they're not looking at the objects directly, they're thinking about them. And what he found, I'm showing you just in a couple different views, a region of left anterior temporal cortex, not all the way up at the pole, and you can see it in a few different views here, of areas that can show above chance classification of the 4 concepts. Now what's interesting, and this is the transformation part, this area does not pass that generalization test for the features. In other words, the neural patterns that are evoked here have some transformation of the information such that they must be sensitive to things like color and shape, because they're distinguishing, and other properties, they're distinguishing among the 4 different fruits and vegetables, but not in a way that simply lets you take the patterns that do color discrimination for one shape and now generalize to the other shape, right? And that's what I mean by transforming the information. Then what Mark did is he asked, what's the relationship between what's going on in the neural codes that are evoked in those part areas in the back of the brain and what's going on in the neural codes that are evoked in the anterior temporal lobe. And to look at this relationship, what he did is on every single trial, he could say, can I successfully, someone's thinking of a carrot, does my color classifier successfully know it's thinking of something orange? Does my shape classifier successfully know the subject's thinking about something oblong? And does my anterior temporal lobe classifier know the subject's thinking about carrot? And he asked what the relationship was on that trial simply by using a regression model where he tried to predict, he did predict the success of the classifier in anterior temporal lobe from whether or not the color classifier was successful. Yes, it's orange. Whether or not the shape classifier was successful. Yes, it's oblong. And whether or not they were both simultaneously successful. And when you look at that model, it's this latter predictor where you see a relationship. Okay? In other words, the simultaneous evocation of color and shape information in those part areas is predictive of this anterior temporal lobe pattern classifier succeeding. But again, remember that information is not the same. It's not simply redundant. It's been transformed in a way that some of the properties of these posterior areas are gone, namely that generalization ability. There are a number of other analyses that I would normally talk about at this point. I'll refer you to the paper and to Mark just in the interest of time. But there's some interesting analyses looking at individual differences that start to give us hints about how people are doing this. Again, this is just to give you a taste of some directions that we're moving and trying to think about what it means to say these features become integrated with one another. The one other thing I just wanted to mention at this point is some older work that we did in my group having to do with what kinds of concepts would be good candidates for this kind of integration. And this is by way of disclosing that we obviously didn't pick tangerines and limes and carrots and celery randomly. Fruits and vegetables are a particular class of objects that are defined by the integration of shape and color information, right? So you need to have both of those sources of information to be able to discriminate them from one another. And so it's not the case that for any object you might think of, you would see this particular pattern. And so a former student of mine, Nina Shu, had done some work where she was interested in the difference between concepts like lemon and lime versus, let's say, stop sign and yield sign, where for both of these pairs of concepts, you know the colors of them. But in the case of, for example, the stop sign, the shape alone is sufficient to discriminate it from the yield sign. But in the case of the lemon and lime, you need both of those sources of both of those features, the color and the shape. And so she did an experiment to look at the difference for the neural codes that were evoked for concepts like this versus concepts like this. And she did this with artificial stimuli, where she taught people new concepts. And for one group of subjects, they learned a world of concepts where color was diagnostic. So the WEMP, the CLARV, and the THUL all are the same shape, but they differ in color. And then a different group of subjects learned concepts where the WEMP, the CLARV, and the THUL had a particular color, but they differed in shape from all of the other concepts. And so the color, although they knew it, they didn't need to use that information in order to discriminate it from any of the other concepts, like the stop sign and yield sign. And then she scanned people after they learned these while they thought about the shape. She had them think about the shape by asking them questions that get at the shape. None of these questions gets at the color at all. And then the question was, would she see activation in areas associated typically with color knowledge differentially in the two groups of subjects? And she did. This is the figure I showed you before. So this is an area that responds when people are retrieving color information. And this is the area that Nina found an interaction between when people were retrieving shape information versus doing a baseline task, differentially active in those two groups. So this area, this color area, is responding when people are making shape judgments. More so for subjects where shape and color were jointly diagnostic, okay? And this, again, gives you a different picture of these feature areas, right? This information is being integrated in a way that you can't just say this is purely color information anymore, right? And so, again, I think this whole breaking the brain into all these parts and breaking the concept into all the parts, you miss critical pieces of the story like this. All right, what I want to do now is turn to one study, again, that we just published, trying to look at the integration and transformation of information, a visual feature with an abstract feature. And this is work that I did in collaboration with my colleague at Penn, Jeff Aguirre, and along with a very talented research assistant at the time, Andrew Persichetti, is now in graduate school with Danny Dilks down at Emory. So Andrew had come to my lab from, he had worked previously in a lab that was interested in decision making. And so I said, what would you like to do in the lab? And he said, I don't know, something related to how people make decisions. And I said, okay, well, I have money to study color adaptation and shape adaptation. So let's try and think of something to link these two interests up. And this is the result of that collaboration. So Andrew taught subjects some very simple concepts. So they're just these moons, moon stimuli, so just these closed contour shapes that vary in color, and they vary in their shape in terms of kind of the thickness of them. And subjects they didn't learn names of them in this case, they learned values of them, but they didn't learn them the way I'm showing you the values. This is just for your convenience. They learned about them by playing a little game with them where they could acquire money. And as they acquired money in the game, that translated into a bonus they were getting for being a participant in the experiment. Now, we assigned every moon a value, an expected value, and we did it in such a way that for any pair of stimuli here, we could say, okay, these two moons here are the same in color, they're different by 1 step in shape, and they're different by $2.5 in their expected value. And we can do that for every pair, so these here are different in 2 steps in color, 2 steps in shape, and 2.5. And we assign these values such that the differences, or on the flip side, the similarities in those 3 dimensions were orthogonal. Okay, and that's going to be important. So we can, for a given pair of stimuli, separate out the effects in the neural patterns of similarity on shape from similarity on value. All right, so subjects learn those values by they would see a pair of the stimuli and they would pick one, and then they would be told, you just lost $7.66, where this $7.66 was a draw from a Gaussian around the expected value of, for example, a negative 750. Okay? And this would go on and on. They'd be accumulating their bank over the course of a day, and then they did this over 4 days. And you can see that they asymptote. They're never going to be at 100% correct, because since we're drawing them from a distribution that are slightly overlapping, occasionally they'll pick one that they actually lose money on. But so you can see that they, by the 4th day, have learned the values of these stimuli, and then we put them in the scanner. And when we put them in the scanner, we are not having them do value tasks at this point. We're having them do what turns out to be a very difficult perceptual discrimination task, where they have to decide whether or not this line that almost bisects the moon is putting more of the center of mass to the right or the left side of the line. So that's what they're doing when we collect the neural data. Okay? They do this before learning and they do this after learning. And then we've arranged the sequences of doing this task so that we can measure what's the neural pattern evoked by this as a function by the preceding sequence of moons. And so we can therefore measure what's called Fmri Adaptation, namely how much this pattern is reduced by the previous stimuli as a function of the similarity of those stimuli to this one in shape and the similarity in terms of value. Okay? And then the last thing you need to know is that we thought about value in 2 ways. So the values ranged from -10 to +10. And so at some level, -10 and +10 are the most similar. The moon that's the -10 and +10, because those are the really important ones, right? If you're gonna maximize your money in this game, you really need to know those. Okay? But in some sense, they're the most different, right? So +10 is the best one by far, and -10 is the worst one. And so we could look at the effect of value similarity, either taking the absolute value of value, right? So -10 and +10 would be maximally similar. I'm going to refer to that as the unsigned value. Or we could say they're maximally distinct. And again, in terms of Fmri adaptation, the implications for this are that if they're maximally similar, the neural pattern evoked by this stimulus, if it's the +10 stimulus, should be lowest when it's preceded by the most similar valued stimulus. So either another -10 or a +10. All right, so let me show you what the data look like. So what I'm showing you here are the effects of similarity, so how much we can predict the response to a stimulus based on the similarity of the preceding sequence of stimuli in early visual cortex. So this is a probabilistically defined sort of V1, V2. Lateral occipital cortex was areas that thought to be coding object information. And then 3 areas that were anatomically defined based on a meta-analysis of value Fmri experiments that was done by Joe Cable's group. So dorsomedial prefrontal cortex, ventromedial prefrontal cortex, and the striatum. Okay? And so the higher the bar, the more that area's response to a stimulus is affected by similarity on that dimension. Okay, and that's again taken as an indication that, the population code there is sensitive to that feature. So before learning, what we see is the darkest bar is the shape effect. We're seeing that the similarity of stimuli on the shape dimension affects the response. Okay, in visual areas and also in some of these other frontal areas. And again, this is while they're doing that shape task, right? So they're doing that task where they have to make the bisection judgment. We're not seeing any response to value, which is good because they haven't learned values yet. So that's just a reality check that our subjects don't have ESP. All right, what about after learning? So I'm just zooming in here on early visual cortex. Here you can see what the mask looks like in lateral occipital cortex. And we have the shape effect. But now we have a value effect after learning in V1. And here I'm showing you the whole mask, but when we break it down in the paper we report, then again, based on probabilistic maps of the different areas, V1 separately from V2, separately from V3, it's in V1. So V1 is a visual area, right? That is the visual area. But what we see is that after 4 days of learning the value of these stimuli, that the neural response has changed such that two moons that are more similar in value have more overlapping responses. Thus, they show a diminished Fmri response or a bigger adaptation effect. Okay? And furthermore, we're seeing it for the signed value. In other words, -10 and +10 are maximally different. And that rules out an attentional explanation, right? Because those are the ones you have to pay attention to, yet they look maximally different from one another. So this is value qua value, we argue, right? This is value in V1. And so I was very, very surprised by this result. And again, I think kind of like with the work that Nina did with the novel objects she taught them, this suggests that information is being integrated in concepts as we use them, right? So we have to link these things together. We can't just decompose them, even in an area that is quintessentially the decomposed region, right? V1, it's vision. Or maybe it's value. We did not see this effect in lateral occipital cortex. We did see shape responses. You know, I suspect this might be because these stimuli don't look like three-dimensional forms. They're just these flat patterns, and we have some ongoing studies where we're using more complex stimuli. So we don't see it there. We don't see this value effect during this task in the value regions. So that's what I'm showing you here. At the very end of the experiment, we had subjects do just one run of a value task. And then we do see some responses here. I didn't bring those data, but they're in the paper. But this, I think, suggests that this is not some kind of, online, even though it's not a value task, they're thinking about value anyway. They're doing this hard visual task, but, they just did this value task for 4 days, so of course they're thinking about the value. Not in a way that we can see here, even though we can see value effects when we tell them to think about value. So I think that's another interesting aspect of the data, an advantage of using this perceptual task. All right, so just to sum up here, what we're seeing is that newly learned concepts after just 4 days, and again, these concepts, they're pretty simple. People know just a few things about them. They know their shape, they know their color, they know what they're worth. But we see that that has transformed the neural responses such that value information and visual information have become integrated in what is typically described as a visual region of cortex. All right. I don't have any idea when I started, because I know it was late. How much time do I have left? 10, okay, perfect. All right. So what I want to do now is turn to this new line of work that we've begun to pursue, looking at not how shape gets combined with color or how shape gets combined with value, but how concepts are combined with one another. And the first experiment I'm going to describe to you involves taking two nouns, so nouns that refer to different concepts, and asking subjects to think of what concept might be described when they're combined. So this is referred to as nominal combination. And the example that I want you to combine is if I said to you that as a result of climate change in the Pacific Northwest, robin hawks are endangered. And you think, robin hawk? what's a robin hawk? And so if I asked you to describe to me what a robin hawk is, or to imagine or draw what a robin hawk is, what I can predict based on data of other people, and now lots of preliminary data we have, is that there'd be roughly half of you who would come up with one kind of description, and roughly half of you that would come up with a different kind of description for the stimulus robin hawk. So here we have a robin, and here we have a hawk. These were drawn by my daughter, who's now being paid on the grant to draw more stimuli. And this was work that was the dissertation of Christine Boylan, and all of this work was done in collaboration with my colleague at Penn, John Truswell. Okay, so now you have a robin in mind, you have a hawk in mind. What's a robin hawk? Well, some of you will be imagining a bird that has some of the features of a hawk and some of the features of a robin. So maybe it's a hawk with a red breast. Okay? Others of you will be imagining a hawk that doesn't resemble a robin in any way, but instead a hawk that hunts. It has it particularly predates on robins. When my daughter first sent me the drawing, it was a much more cheerful looking robin. I said, no, make it clearly dead. So there's version 2 of the robin hawk. All right, and so we and other people have argued that these are qualitatively different kinds of combined concepts. What you've done here is decompose, recompose, right? So you've taken your concept, you've broken it into some parts. Through some properties that we don't understand, you've decided which parts to pull from the robin and which parts to pull from the hawk, and you put them back together, voila, new concept. Here, no decompose, recompose, right? So here what you've done is you've taken your concept of hawk and you've taken your concept of robin and you've put them into some event structure. So there's this implied predicate now of hunting that you brought to the table, right? And so now your robin hawk has robin and hawk in these two different roles with respect to one another, but you haven't done any of this decomposition of them. All right, so what we've been interested in are the neural systems that support these different kinds of combination. We're interested in understanding what features are taken from one and put to the other by looking at how the neural patterns are changed. We're interested in Sarah Solomon, who's here, has been doing some work looking at how, not just in these nominal combinations, but how in metaphors of the sort that Anjan talked about earlier in the meeting, how those are examples of conceptual combination that change the concepts. And I'm going to show you, if there's time, a new direction we're looking at in how the very process of recombining these concepts actually changes, at least briefly, your conceptual system. All right, so let me show you an example of how this work proceeds. So I was on sabbatical at Princeton a number of years ago, and together with Dan Ascherson, we did a study that, at the time I didn't really know we were studying conceptual combination. But what we did was we had people monitor a series of faces, and they were either given the instructions to look for the old faces, or look for the women, the female faces, or to look for the old women, and so on. And then we could say, what are the neural patterns evoked when people are thinking old and people are thinking woman? And is there some combination of these two patterns? And it turns out the combination was plus. We add the two patterns to one another, where we could predict the pattern when they were looking for old women. Okay? And it turns out there was one place in the brain where we could successfully do that, where we could combine the neural patterns to get the pattern from the combined concept. And that is this region in the left anterior temporal lobe, not quite at the pole, but up on the lateral surface. So we wrote the paper and we talked about this, areas being involved in conceptual combination. What we didn't do was talk about this as being a kind of attributive or property-based combination, which is the name the field uses for describing the kind of robin-hawk combinations where you have the hawk with the red breast. Okay, so you have some property that you now are applying to the other concept. But that's what this is, right? This is like decompose, recompose, right? There's other work that's been done. Jeff Binder, his group has been doing a fair amount of work looking at conceptual combinations and finding responses in the angular gyrus, sometimes on the left, sometimes on the right, sometimes bilateral. When people are doing conceptual combination, I'm going to actually skip a background study just in the interest of time. So we hypothesized that the anterior temporal lobe and the angular gyrus may play different roles for these different kinds of combinations. And so in a study that is forthcoming in Brain and Language, we had subjects combine nouns that were strongly biased towards a relational kind of combination. So this is like the hawk that hunts robins, versus an attributive combination. Robin hawk, by the way, is a non-biased one. That's why I know that 50% of you would have one and 50% would have the other, particularly if you're a native English speaker. But these are strongly biased, and we had people combine them. They were asked to make a judgment on them, but again, we analyzed the data from this in-between time, and we looked in regions of the angular gyrus and the anterior temporal lobe at the responses, and we didn't find a straightforward double dissociation. Instead, what we found was that the angular gyrus showed some specialization for relational over attributive, but in the anterior temporal lobe, we saw what looked like a timing shift. Okay, and so we don't know what this means yet. We don't even know if it's real yet. Fmri is not the best method for looking at timing changes. But we're now trying to design studies where we can look more closely at these patterns. Okay, so that's one direction that we're going in this. I mentioned that we're interested in the consequences of doing this combination. And so I have a new postdoc in the lab, Yod Kennett, and he came to me with a very strong background in applying these graph network analyses of semantic networks. And so what we have is we've modeled the semantic networks. So this is, each of these nodes refers to a concept. They're drawn from different categories. We do this before and after, so separated by a week or so. Before and after subjects on the second day do either attributive combinations or relational combinations. And we're going to be presenting the first pass at these data later this summer at the, he's giving a talk at the Cognitive Science meeting, if any of you go to that. But looking at different changes to the semantic networks in between this week where they did this combination as a function of which kind of combination they did. And this is just one example of the results, where if you look at how similar the networks are to a sort of a priori network defined based on the category structure. So I mentioned these came from these 5 categories. So what you see is that decompose, recompose changes that structure. And doing the leave them whole and put them into an event does not. And again, because we're really interested in how dynamic this system is and how using concepts really is the question, right? I'm very excited about this direction. I mentioned that this relational stuff is sometimes left, sometimes right. That's something else we're very interested in, because when I introduced the idea of having the robin and the hawk, and now they're in these roles around an implied predicate that's an event. This is an area that, for people studying events, is the angular gyrus is a likely candidate to be involved. They'd be described in very different kinds of ways about actions and so on. But it's also an area that turns up in studies of verb processing a lot. And in a totally separate line of work, Christine Boylan, again with John Truswell, we did a study where we were looking at 2-word phrases. So these aren't nominal combinations anymore. So things like eats meat, eats quickly, with meat, tasty meat. And we asked, for any given pair of these, you can say, are they similar in terms of the noun here? Are they similar in terms of a couple of linguistic distinctions that are made? And when we looked at the pairs that were similar in terms of having a common verb, so eats meat and eats quickly, we saw one area where the neural patterns similarity reflected that similarity, and that was the angular gyrus. Skip over that. So when we have eats meat and eats quickly, we see a similarity in the neural patterns that are evoked in the angular gyrus, but this was in the left. The previous result I showed you was in the right. And so we think that maybe this is a coincidence, but maybe there's something here about the difference between an event that's represented in this implied way of the robin hawk versus one that's lexicalized here in eating. And so that's something we're interested in exploring. All right, so come back in 9 years if Tom Griffiths is on the schedule. You might get him, you might get me, and I can give you an update on some of the things that we're doing. And I mentioned there's been a lot that's been going on. I told you Sarah Solomon's been doing some work on metaphor comprehension. We've been doing some work looking at how priming can dynamically reshape the network. And again, this is a general question that Sarah's been really interested in. Another thing that we've been exploring is how the variability in neural patterns that most people average over to get a stable multivoxel pattern, how that variability actually contains important information about semantic variability or contextual variability. So it's throwing out that variability. You're actually, again, missing an important part of concepts. And we've been looking at different consequences of how people learn new conceptual information. So that's what you'll get 9 years from now. And so for now, let me just end by thanking Mark, Andrew, and Christine, who really spearheaded the work I talked about, thanking Penn, in particular the Center for Crime Neuroscience, and also my funding sources for supporting it, and thanking you all for your post-social banquet attention.
Okay, we have Alex here.
So who talks faster, me or Uda?
Excuse me?
Who talks faster, me or Uda?
Uda wins. No, you were actually much slower than usual.
Yeah.
So my first comment is, wow. I mean, that was fantastic. So I've been getting very pessimistic about, seeing any further success in this area, any forward motion. So I'm feeling much more optimistic. I thought it was terrific. So first, a historical note. As you well remember, the very first study looking at semantics, which was published in Nature and had tremendous influence, showed data and made the argument that semantics was basically a dot in left frontal cortex.
Yeah.
So when we came along, and then other groups, right, there were three important findings. One, if you looked at knowledge across different kinds of features, like colors and actions, you got activation in different places, wasn't one. That alone could have been totally uninteresting, because as Alfonso used to say, what difference does it make where the lesion is, right? It's going to be in different places because these are different functions. But the other two important things were, one, where the locations were seemed to make sense based on our understanding of the organization of sensory and motor and affective systems. And then third, at least in our hands, it never, ever, ever was in primary cortex. We never saw activity in V1 or M1 or A1. It was always in these kinds of downstream areas, and that led to, what is the relationship between the two, as you pointed out. So you showed several studies where you get this anterior temporal area, just now the angular gyrus, but the V1 finding is particularly provocative. So my first question is, did you actually anatomically, were you looking just at V1 for that? Or because what you showed was that big occipital.
We did in the paper we reported a further parcellation, again using these probabilistic maps. We also had retinotopic data from these subjects doing the standard rotating wedge. So we have both functional and anatomical. I think it's the anatomical stuff we report in the paper.
So
yeah, it's a V1.
That part went fast, but how did you rule out the attentional explanation?
Rule out is strong, but I went into this thinking that what was likely to happen in visual cortex is that we would see the most adapted response for stimuli that were the most extreme, so the positive 10 and negative 10. So that if you saw a positive 10 moon and you had just seen either a positive 10 or a negative 10, because those were the moons you had to pay the most attention to, you would have, for each of them, a heightened response, therefore more of adapted response. And that's not what we found. So positive 10 and negative 10 in sequence produce the greatest response, that is the least amount of adaptation, which the inference then is the most separated codes. Okay, so that was the evidence against the attentional account. Yeah,
So now how are you, so one idea, if this is kind of a feedback phenomena, which I assume you think it is, you might have predicted, well, certainly not V1, but maybe more object-related visual. So how are you thinking about this V1 business?
We keep seeing stuff in V1. So there's another paper we published last year. Nick Hindi is the first author, actually a series of papers in collaboration with Jerry Altman, where we're looking at when people, when an object changes in the course of an event, how the neural patterns that are evoked by that object track that change. And so one of the paper's subjects did a task where they imagined, for example, a wine glass, and then they had to imagine breaking the wine glass, and now they were told to imagine the wine glass, either before or after the event they just imagined. This is all imaginary thoughts, real data, and we ask, where do the neural patterns track the similarity of the whole wine glass versus, am I allowed to break this? No, I'm kidding. Versus the broken wine glass, right? So the wine glass transformation is a big one. Opening a door is a small change, right? And so we ask, where do the neural patterns track that similarity? V1, in their imagination. So, you know, why didn't we see these things before? Well, it's a totally different tool, right? It's not like, oh, we're asking, are we evoking a V1 response? Now, in the case of the moon study, there is a visual pattern. In the case of the wine glass study, it's just in their imagination. But in that study, we did not see the neural patterns in lateral occipital complex tracking that change. Sarah, when she first joined my lab, did an analysis where she showed that the LOC patterns track the similarity of the wine glass to the door. So they responded to the overall category of wine glasses, visual similarity as related to doors, but they didn't care about the particular instantiation of that wine glass, right? And maybe that's the difference between what's going on in these different codes.
I have a very related question on that same study. Thinking about the results in V1, it reminded me of a growing number of studies by people like Lars Mukli and Petra Vetter that show that you can decode surprisingly high levels of information in early visual cortex, like different sound categories, for example. And they followed this up with TMS, showing that if you zap V1, actually their performance is reduced. I'm sure I'm not the first person asking you that. So my question is, has two parts. First, have you considered applying TMS to V1? And second, you only showed this result in V1, but I'm wondering, did you do a whole brain analysis to see whether you find this reward or value-based adaptation also in other areas other than V1? Because that would help you to interpret those results.
We look throughout the visual system, and the effect diminishes as you go upstream in the visual system. And we looked in the value regions, anatomically defined. We didn't functionally define the value regions. And we, as I mentioned, well, I did it very quickly, but we did not see any evidence of value coding during our perceptual task, which is the task I showed you, but we did see evidence of value coding during a value task, although that was a very underpowered test because it was about 8 minutes of data. It was just a tack on at the end, a very, it turns out, useful tack on. But so that all needs to be followed up in another experiment. What we're doing now is we've collected a whole bunch of data. This is work done in collaboration with my colleague at Penn, Danny Bassett. We've collected data while subjects are learning. Again, they're a more complicated set of stimuli. They look more like the novel three-dimensional rendered objects that Nina used in the work I mentioned. And subjects are learning the value. In this study, they're learning it in 2 different ways. So we're manipulating kind of how declarative versus procedural the learning task is. But we're scanning them on 4 consecutive days of learning to try and look at how information, how the networks that are coding visual information and value information might become integrated in time, and whether or not when you see integration using some coherence measures, again, this is all stuff that Danny Bassett's developed, whether we can predict when subjects learn based on normally segregated or modular networks, so networks that have different community structures, whether we can see a decrease in modularity and increase in integration that's related to learning. She's shown some of these kinds of effects during a motor learning task, but never something like this. So I think then we'll have a handle on other parts of the brain and how they're involved, because that specifically is a whole brain network analysis during the learning over 4 days. It's a lot of data.
Just as a quick comment, maybe what you just said about Lars Mugli's work and the multisensory thing, and also like V1 and decoding performance in V1. So I feel like, I mean, one, there are many voxels. Two, it's a particular retinotopic organization which enables multivariate pattern decoding and helps this. So for instance, like for these multisensory effects, even though you can decode slightly above chance something from V1, and even though perhaps if you apply TMS, you might have a tiny little effect in terms of response times, I'd be very cautious of saying that V1 is really relevant for encoding, for instance, sounds, right? So it's very well known that if you present a sound, you get like strong deactivations in V1. Also like TMS, I think people always claim that TMS, as long as you don't look for auditory things, it's unconfounded. It's of course not the case. As soon as you present like a TMS click, you have a strong deactivation in visual areas again. So in this way, I just want to be, I'm also like a little bit cautious still about like whether visual cortex really has, just because one can decode something from V1, whether it is really so functionally relevant, right? I'm still like wondering whether it might be just some top-down modulation. I mean, we all know it's like more sensitive to LFPs, which might not be reflected in the spiking patterns, etc., etc. So I just feel like with the decoding thing, we get into a new field, which might depend on the organization of vasculature, on the organization of the neurons, right, and so on. So that's, I think somehow we always need to compare also like how well can we decode other things from the same area. And that's why I'm just because you mentioned this with this sort of like Lars Mugli multisensory stuff, and there I'd be a little bit cautious. So that's, and this might also relate also to conceptual to concept being represented in there.
No, I mean I absolutely agree, you know, I was just struck by how, you know, I'm interested in dynamic changes in these networks, and so to see the neural response has changed, right? So there's no question about that. Whether or not it's changed in a way that supports the ability for subjects to make the value judgments, obviously we have no idea. Nor do we know, and I wish we did, but we didn't collect these data from these subjects, we don't know whether we've changed their perceptual judgments, right? So you could say for moons that are now have more different values, what we're arguing is that the visual response is more different, right? So we've changed the response to make it different. And so regardless of how similar they are in shape, we have pulled those neural patterns apart, right, through the learning. Well, that leads to the prediction that they would be better able to make a perceptual discrimination between those moons than if they were similar in value and we pushed them together, right? Because we didn't think we were going to see these effects in V1, we didn't collect those data, right? So, but I mean, that is absolutely, you know, I think in some ways I would be more interested in that result than whether or not V1 is supporting the value decisions. I'm interested in how learning the value of something might change our perception of those things. That's true, yeah. That's right. When subjects were doing a value task, we saw responses in value regions that reflect value. What we argue in the paper is that when they're doing a perceptual task, they don't recruit those areas because they're not thinking about value. And those are areas that are very task specific. I mean, in the value literature, they're all explicit value tasks that people are doing. Those areas respond in ways that reflect the task demands. And so they show shape adaptation effects, we hypothesize, because they're doing a shape task, right? And so that's what's rewarding them. Getting the shape task right or wrong, it requires attending to the shape. So that's our, I mean, we don't know for sure, but again, we had the perception task, and then we had, which was very powered, because to do the Fmri adaptation, we needed a lot of data to hit all of the different steps of similarity an equal number of times. To do the value task, we just have a few minutes of data, what was left at the end of the 2 hours. So.
Okay, I have four questions lined up. One here, there.
I have a question regarding the first study in which you decode color independently of shape.
Yes.
And I was wondering, so typically and unfortunately, dimensions are often intercorrelated and correlated with other features of no interest. And in your example, I was wondering how sure you can be that you really isolated color independently of features like, so you had fruits and vegetables that were orange and green, and orange might be more associated with sweet or ripe, and other ones like...
Yeah, so I mean, it's an excellent question, and we for sure can't isolate them perfectly. But let me go back to the design. So the way we analyze the data is we train the classifier to make this discrimination, okay? And so we're going to call that a shape discrimination. But that could also be a phonological discrimination. That could be the discrimination between someone internally voicing tangerine versus carrot, okay? So we could have a classifier that's picked up on the tangerine sounds versus the carrot sounds, and that would succeed, right? Absolutely. Now we set the weights of the classifier, and the test data that we use are not more tangerine and carrot trials. They're the lime and celery trials. So if there were parts of that classifier that were succeeding because they were coding the phonological properties of tangerine, carrot, they should not be able to predict whether someone's thinking lime or celery. Okay? So the generalization test rules out a lot of confounds, but not all of them, right? I think that's the power of that test, is showing that it can generalize across. We can take this classifier and generalize to this, and then flip it, this classifier, and generalize to this. So in the maps that I showed on the slide after this, those are just the regions that pass this generalization test, which is much more stringent than had we trained the classifier on all but one of the runs of these stimuli versus these stimuli. Right? That's what most people would have done. That's what I would have done. But Mark was smart enough to realize, no, there's too many other differences between this and this. Okay?
Still, I mean, orange fruits and vegetables are more sweet, and ripe, and green ones are maybe not ripe, and this could be a quite important dimension. I was wondering, is color evolutionary speaking more important than such dimensions, or from semantics? This might be a more important dimension that you actually decoded there.
Absolutely. And for the purposes of the story here, it kind of doesn't matter, right? So let's just say it's the sweetness. Let's say it turns out V4 codes sweetness. Okay, so it's getting sweet versus bitter. I'm happy with that. I don't think it's true, but I'm happy with that. Because what I've shown is an area that is representing some properties in a way that it can generalize across variation in another dimension, and that the simultaneous evocation of that information alongside some other dissociable kind of information is predictive of concept decoding in the anterior temporal lobe. And it doesn't need to matter what it is. Now, I think given, and this kind of gets to Alex's point of where the things are and make sense, I'd be surprised if it was gustatory, but it actually doesn't matter for the story. So you're absolutely right. It's an important point that we don't, when you use these real world stimuli where there's all this stuff you know about them, you're not going to be able to control for everything. But I think the generalization test helps with a lot of that. Not all of it, but a lot of it.
I think we're jumping around a little bit on you. Apologies for that. I'm going to move back to the value data as a separate topic there for a second. So in your response to Alex, I think you kind of addressed some of what I wanted to ask about, but I was also surprised that you interpreted this as the absence of attention. And there's been a lot of work on this recently, particularly with time-resolved measures like animal physiology and these other, that it really looks an awful lot like attention. What actually ends up happening in V1 in particular, when you look at animal data, it looks like what's happening there is a variation in how attention is encoding value in some sort of way. And what might be happening in your data is that you have loss aversion in this attentive response. And I wanted just to hear you kind of comment on that. If you think that that's a reasonable interpretation, you're interpreting this as a very strong value representation in some sort of way. If it were still attentional in nature, where you had some of these decision-making principles acting on attention, could that also account for what you're seeing there?
Well, so let me just back up a second and say, absolutely there are attention effects in V1. So I would be ridiculous to say to anyone that there are not attentional effects in V1. So absolutely. And the question is whether or not attention is the explanation for the changes that we're seeing in the pattern. And I don't think it is. Even if you have a negative 10 and positive 10 are not identical because of what we know from the decision-making literature about negative 10 looms larger in your mind than positive 10. So negative 10 is more bad than positive 10 is good. That's I think what you're referring to. So then they're not identical. What I'm saying is that the Fmri response indicates that they are maximally different from one another. That negative 10 produces the least response. The negative 10 moon shows the smallest response, the smallest adaptation, the biggest response when it's preceded by positive 10. And it's a very linear looking effect. It's absolutely monotonic. Linear is hard to say with Fmri data, but it doesn't have the properties that you would see a non-monotonic effect along some old pictures of my kid. These are also from about 2007 or 8. I don't know why that just happened. All right. So it doesn't look like, we looked at the data for exactly that to see whether or not you would see what looked like kind of a alteration at the edges. But I still think you would predict, unless I'm not understanding the loss aversion prediction you're making, I still think you would predict something non-monotonic as you moved from negative 10 to negative 7.5 to negative 5 and so on. Okay.
So my question was, that's a great lead-in, because the puzzling thing for me about the V1 result, what would you think would happen if you were to change the task, just do a behavioral measure, and say, all right, it could be that negative 10 is really salient and positive 10 is really salient. Maybe those are equally salient, and during learning that really boosted sort of your processing of those things. I think you would predict, if you had some fast speeded response time task, you would get a U-shaped RT curve, right? Based on rank eroding by value.
Yep.
But it could also be that asymmetry with positive versus negative would really boost only one side of those. So, the interpretation is, that it couldn't be an attentional effect because both of these things are equally important. But we don't really know the nature of the change, whether or not, there's lots of talk in the attention literature about salience maps and things like that. It could be that you actually have more salience for just the positive things.
Yeah. So
in which case the RT thing would be asymmetric.
Right.
But that would just be a behavioral way to assess whether or not, when people are doing a task that doesn't have anything to do with value, with these exact stimuli, whether or not it is manifesting in that.
Yeah, well, and there are data showing that. So the effect that I said I wished we had in my subjects, and Sue said you can get an undergraduate to do it, other undergraduates have done it, right? So when you teach people categorical distinctions between stimuli, it changes their perceptual judgments. And I think what you're describing, you would see differences in response time of the sort you're talking about. All I'm saying is I don't think those differences are the basis for our adaptation effects.
Right. Well, so just two more little follow-up thoughts on that. So if you were to rank order the visual areas by the magnitude of the attentional effects you get, V1 would not be the first one you would think of, right? It would be, that's not where attentional effects are the largest. In fact, it's pretty hard to get a big attentional effect in V1.
Well, I think it depends on
relative to other areas.
It depends on what attentional effect you're talking about and what task you're talking about.
Right, but just visual selective attention, that's why people study V4 in monkeys. It's harder to get the effect. The effects are smaller in magnitude. So that part of it's kind of weird. The other part that's kind of weird, though, is if you just look at visual adaptation studies, V1 is also not the first area you would think of about areas that would show a visual adaptation effect. You often don't see anything in V1 when you do at least with objects, you're repeating objects. That would be an area that would actually typically show something smaller, even maybe a negative response. So
yeah, there's a very nice paper that I'm some 18th author on or something, but from Jeff Aguirre's group that actually looks at the kind of time constant of how long you're adapting over, and it seems like that varies throughout the visual system. So I really think the details are in some of the task. I don't think you can make that statement without putting a lot of qualifiers on it depends on the task.
It is a bisection task that you're having them do. So maybe in that particular context it changes it so you're really emphasizing V1.
Right.
Okay, we have time for 2 more questions. One here, one in the back.
So I was interested in an effect you put up very briefly, and let me remind you what you said, and because I didn't understand what the measure was that supported that, and then I have a question. So I was really interested in your, I forget which of the 3 pieces it was, but your noun-noun combination experiments and the relation to actual syntactic combination in general, because of course for many people in cognitive science, what a theory of concepts needs to explain is conceptual combination, not primarily categorization.
Right.
So I love the direction you're going here. And so what you showed was that if people are doing robin hawk in terms of a hawk that eats robins, you don't see evidence for a change in the representation of robin or hawk. But when they've done robin hawk as a hawk with robin properties, you do see some reflection of change someplace in the actual representation of hawks or whatever it was. So could you tell me what changes those were?
Yeah. So we are trying to do what you're describing. We haven't done that yet. So so what I showed you is much less interesting than what you're describing, which is what I showed you is that in the right angular gyrus, we see a greater response when subjects are engaged in the relational kind of combination, so a hawk that hunts robins versus attributive. But what we're interested in is what it means to combine those 2 kinds of information. So what I showed you in the old woman experiment, this one, is that in the anterior temporal lobe, an add function is a way that you can combine the concepts, right? Or it's the same as an averaging function. But that won't work for a relation, right? This should not work in the angular gyrus if the angular gyrus is coding relational information. But there actually isn't a good model. There is not a good theory of how that is combined in the robin hawk case. What we do know, this is not a statement about the brain patterns, this is a more interesting statement, probably about behavior, is that if you have subjects make a series of these judgments in a row, you get a priming effect if they've just made a bunch of relational ones on the subsequent relational versus if they just made a bunch of attributive. But furthermore, we know that if they're making a bunch of relational ones, if you have 2 items in a row where the relation is the same, you get a priming effect versus 2 relational ones where it's different. So that event is in there, right? And so what we don't know is if there's going to be a way that we can discover the process or understand the process by which these are combined by looking at the neural patterns. Maybe not, right? I mean, maybe just doing these kinds of experiments is the way to go. But it's not going to be this. Right? And so, and I think that's our fundamental claim, is that these are just qualitatively different ways of combining information, and they both happen all the time, right? So we're doing both of these all the time. This one, we have, Sarah Solomon has very interesting data that she very nicely sent me when I gave her 36 hours notice that I was giving a talk, and then I couldn't get email in my hotel room. So I'm not showing you her data, but looking at how when you have an adjective and a noun, how those combine, but how it depends on whether or not that noun that concept already carries information, like the way that we don't say yellow banana, right? The yellow goes without saying. So when you give people yellow and banana, you don't see the banana patterns change in the same way that if you say brown banana, right? And so she can show that the amount that those patterns change is predicted by what we know about bananas. Did I get that roughly right? Okay. So it's add, right, for the featural kind of combination, but it's add with an asterisk that it depends on what you already know about these concepts, right? So it's not this simple, although adding did a fine enough job. But for the relational ones, it's not add, right, in the same way. So that's what I think is super interesting. Cool. And by the way, not all languages can you do this with two nouns. So there's some languages where you stick two nouns next to one another, and it invites only one interpretation. But obviously, your conceptual system can do both kinds of interpretation, but you just aren't going to do it with two nouns.
Thank you very much. I think this was the end of the morning session, and we're going to go for coffee. We'll come back here in half an hour.